All right. Good morning or good afternoon, depending on where you are. My name is Gabriel Lopez. I'm professor of chemical and biological engineering at the University of New Mexico. And I'd like to welcome you to our SynCell 2021 speaker series. Today we have two talks from Germany. The first one will be by EBERHARD Bodenschatz, and the second will be by Lado Otrin. I will introduce Dr. Bodenschatz here momentarily. I just want to remind everyone that we encourage you to submit questions through the Q&A feature on the webinar. And at the end, we'll get through as many of those as we can. Basically, we have a talk for about 30 minutes by Dr. Bodenschatz, and then we'll have about 10 minutes for questions. And I'll try to keep him to that time. And then we'll switch to Dr. Otrin. Okay, so we are very pleased to have Dr. EBERHARD Bodenschatz speaking to us today. To my knowledge, he started his independent academic career at Cornell University, where he rose through the ranks to become a professor of physics. After that, in 2003, he became director and scientific member of the Max Planck Institute for Dynamics and Self-Organization, and he continues to have close ties to Cornell University, where he is adjunct professor of physics and mechanical and aerospace engineering. In 2007, he also was appointed full professor of physics in the George August University in Göttingen, and he is also co-director for the Max Planck University of Twente, Center for Complex Fluid Dynamics in the Netherlands. And finally, I would like to mention that he is a founding and steering committee member of the Max Planck School Matter to Life, which is focused on engineering of synthetic cells. So with that, I will let Dr. EBERHARD Bodenschatz take it over to talk about synthonemes, building blocks for artificial cilia and flagella.
Well, thank you, Gabriel, for having me. So on my talk, I will summarize our recent work on synthonemes. This work is done by Isabella and Azam Gholami. We're both group leaders in my department, and I'm mostly reporting their work. But I hope I can answer most of your questions also. And so I got fascinated of cilia a while back when we investigated together with Gregor Eichele here from the NPI for biophysical chemistry, cilia in the brain. And here what you see in this video is a cut through a tissue in the third ventricle of the brain. And you see these cilia beating along. And this is what's happening right now in our brain, hopefully, otherwise you would be sick. And so the cilia, they're typically 10 microns long. They're cells. These cells are multiciliated, so they have many of these little hairs that are beating. And of course, the flow that is generated by these hairs is mostly in a very viscous regime. The length scales are tiny, the flow velocity is so small, and so the Reynolds number is also very slow. So this is in the highly viscous regime. And so I have to, there we go. And so as you see here is a summary of a few examples of cilia. One is, of course, sperm, which is propelled by a flagellum. Chlamydomosa has these two flagella, which are beating like this, and the cell is basically swimming along. I will use this one in further down in my talk for quite a few things. There's other multicellular organisms that use this, or we have these trachelial cilia in our lung, which hopefully propel all the mucus containing all these viruses out of our lung and gives them away so we don't get sick. And then here you see a cilium beating in the brain on the way on the right side down there, where I just show you the video. This is by Christian Westendorf here in my institute. And so what cilia are everywhere in the body. Without cilia, it would not be possible to have a baby, not only because of the sperm, but because of the ovum, the egg in the uterus is transported by cilia also. And so there's there's some in the kidney they're everywhere, and the cilia they can be sensing cilia, or they can be motile cilia, and I will only talk about motile cilia. And so in the brain, for example, here you see a little cut through the third ventricle of the brain. And what's most exciting about this is you have these ependymal cells. These ependymal cells have these little cilia. And then down here you see a typical transport on a cutout of this tissue. And so this is about probably 400 microns across, 500 microns across of a millimeter. And what you can see is you can have very distinct transport in a 2T sheet. So what you do is you put a dye down, and then you can see that the cilia lead in a certain direction, and they transport the fluid in a very directed manner. And this you can also read up in this science article that we published quite a while back. And so on the right side, you see they beat very, very fast. The time scale here is milliseconds. So you have a very, very, very fast beating structure. How are these made? What are these cilia actually made of? So they are ubiquitous in nature. So pretty much every eukaryotic cell has it and can make it if it's programmed to do so. And so what you have is you have a basal body. So you have to anchor one of these little hairs, which are called axonemes, and you anchor them in the basal body inside of the cell. That gives them a direction and also a B direction. And then on the top, you have this axoneme, and inside you have these microtubules. So you have 2 central microtubules and 1, 2, 3, 4, 5, 6, 7, 8, 9 outer microtubule pairs. And these microtubule pairs are connected with dynein motor arms. So these are dynein motors, and these dynein motors act on these microtubule pairs and bend them. And through that bending, the axoneme moves, and when the axoneme moves, it can basically whip like a whip, and this therefore transport this very viscous fluid in a way that it basically goes through the 3D space, then goes back onto the surface, pulls back, and then goes out again. And by this, these microtubules the cilia can actually transport fluid. So how is this done? Well, as I said, here are these microtubule pairs over here. Then here you have these dynein motors, the outer doublet between the outer doublets. And then what happens is you give them ATP. And when they get ATP, they somehow get this elastic structure, which is about 10 microns long, to bend. And through this bending motion, these things start to beat and can transport fluid. Now, as I said before, is you have not just 2 of these little arms, you have, I don't know why I can't go back. Okay, I don't know here. What you do is you have these 2 here, there's another pair, and then in between you have these dynein motors, but it continues like this. And actually how these beat is understood to some extent, but not really fully. And what you definitely need is these 2 central microtubules. If you don't have those, then these cells will not be motile. They will just be sensing cilia. And so these are little, basically, nanomachines, and it would be wonderful to reconstitute these things from the bottom up, which we typically do in bottom-up synthetic biology. And so first I would like to talk about the work by Isabella Guido. She builds synthetic axonemes. So axonemes, as I've shown you, were these structures which are inside of one of these flagella. You have these doublets, and then they're beating. And so Isabella set out to build some of these artificially. And she used stuff originally from the literature. And so what you see here is you take these few building blocks, and the idea is you take microtubules, you take kinesin motors, and very much like a sliding assay, which was already known before. And then you ask yourself, can I build a beating structure with a microtubule and a surface? And so what she did is she basically reconstituted the system where microtubules would grow. These microtubules would actually adhere to one end. So it's not quite a sliding assay. There's little tricks that you can do to the surface. So this actually happens so that these microtubules don't slide along the surface. And then you have here these kinesin motors over here. And then these kinesin motors actually transport and bend this elastic biopolymer, this microtubule. And by this bending motion, you get a certain force. And when the force is exceeding a critical force, the motors let go and the whole thing will relax. So let's see how that looks like. So here in the gray that you see in the background here, is the actual experimental observation, and the green and the blue is a fit to the structure. The red are the force arms that one needs to actually reconstitute this motion that you see over here. So let me play it again. So what you can see is this thing is actually being moving around, and then whoosh, you see all the forces are gone. Then it goes straight, then it goes straight again. The forces bend the whole thing until the force exceeds the threshold, and bang, it lets go again. And so this one can actually analyze. So Andrei Vilvan and Rami Golestanian came up with a model which uses elastic beam equations. And then you have the flexural rigidity of the microtubules, and you put in motors. And then you follow basically, this is the gray, here's the experimental observed structure by a fluorescent microtubule. And the green is basically the best fit one can get by using different forces. And then what you can do is by this, you can see what forces do you actually get. You can also ask, are these forces always tangential or are they perpendicular? So are they along the microtubule or at the right angle? And you would see both of them are quite important. So you have a force which is centered around 0, but minus to plus 10 piconewton. And then you have also minus 10 and 10 piconewtons in the transverse direction. So the longitudinal one you would not have thought would exist, but of course it does, because it basically pushes the microtubule back, and then it starts to buckle, and then also leads to this motion. So this you can read up in this paper by Isabella in Nano Letters in 2019. And if you just click on this QR code, you will see the paper. And so this allowed me to build with this Isabella built a very, very simple synthoneme, if you want. It's just one little microtubule attached to the surface at one side, and then motors push it around. But this is not a flagellum. A flagellum, or a very simple axoneme, you would like to have go into the third direction. You would like to have it beat out of plane, anchored to the plane, and beating upward, just like an axoneme that I show you here on the left. So this is what you want to do. And so what Isabella realized is the most simple structure is, why don't we use just two of these dynein arms over here and attach them with dynein motors? If you have these dynein motors, not kinesin motors, but actually the real motors that are being used to make axonemal dynein, to make an axoneme, can we rebuild a beating structure? And so what you see here, so here are these microtubule pairs, or the microtubules, and then you give them ATP, and then they should also beat without having this complicated structure of the axoneme. So how does she does it? Well, what she does is she takes a chlamydomonas algae, like the one you saw swimming at the beginning. You can cut this off with some chemical procedure, and then you can denature this whole beast, and at the end you can have little parts of this axonemal structure over here, the little tiny parts that look like this here on the left side in figure 1. And so then what you do is you then grow, like shown over here, you grow your microtubules out of that structure. So down here is the axonemal seeds, and then out of the axonemal seeds you grow microtubules. Of course, they will grow in any random matter, but once in a while you're lucky and they grow next to each other. And then what you do is you take this outer dynein motors, and you attach them, in a manner that is exactly the same as it's done in the real axoneme. They're attached in a periodic manner with a distance of 24 nanometers periodicity, just like in the natural system. So what you need is a ruler protein that allows you that these guys actually attach in a certain periodic manner, just as they do in nature. And here you see different analysis done. You can also see this in the paper. You can make an X-ray diffraction, and electron diffraction, and you can actually see where these motors are located and what's the periodicity is. So what you now have is you have a two-armed synthoneme. So again, synthoneme is just a synthetic axoneme, and the most simple version of it to be true. And what you then get is you have these outer dynein arms. You attach them in a periodic manner. The distance here is about 30 nanometers. And then, of course, what you can also have is they could be further apart like this, and then the motors are even further stretched, and the distance would be 200 nanometers. So it would be a much larger distance. Now, it's a little hard to really see what you have under normal microscope, but what you get is the following. So with the motors from Japan, what Isabella was able to do was, with her collaborators, she was able to take these. So this is a microtubule over here, and this is a second microtubule, which now has these axonal dynein motors. And what you can see is that you get this constant flexing going on and the bending and slashing in the system. So how can we understand this? Well, let's think about a buckling instability. So let's take a bar, and you take this bar and you push the bar together. Then what you get is you get just a buckling. If you then stretch it a little less, then of course it's like this. Or if you have two of these next to each other and you slide them, let's imagine you have two elastic rods. You clamp one side with your hand and you slide the other one up. What you will see is that the lower one suddenly will let go and will actually buckle out with respect to the one that supports it. So before we had a surface here, remember we had the motors on the surface. Now we actually have two microtubules. These two microtubules are sitting next to each other, and in between are these motors, and they're being pushed like this. And then one side buckles out, and then finally it lets go and it relaxes. So here you see this. So first you have this situation. You have this very strong force between these two microtubules. Then at a certain force, some of the motors will let go. And then you get this buckling, but there's still some motors left over here. And then you get this motion. And when you do an analysis, you get that this force is 90 piconewtons, but this is also in the paper and you can read it up. So how do we understand this motion that is seen over here? So here you have these two microtubules. In between, you have these axoneme motors. These axoneme motors push the upper microtubule, which is clamped over here, towards the other one. It pushes further and further until the critical force is reached and the whole thing lets go. And with this, you can actually explain this motion very, very well. So with this, we think this is the first time that there's an active beating of a reconstituted synthetic minimal axoneme, which I like to call a synthoneme. And so you can find this paper here in the literature, just on arXiv. We are still publishing this work. But I find this work to be absolutely essential, because in contrary to what other people have done, is this the first time that you actually have a very, very simple system of two microtubules which extend into the third dimension, which are actually moving with respect to each other and are beating. And this is only possible because we use axonal dynein motors. With no other motors, we were able to get anything close to that. So it's quite different to where we use the kinesin motors, the double-headed kinesin motors. This is very, very different, which is very close to a sliding assay. This is not a sliding assay. This is very different. And so so in summary, what I've shown you so far, I've shown you this very, very trivial synthoneme, which is really just one microtubule on the surface being pushed around by motors. And here I've shown you the promising result of the first synthoneme. So we can assemble more complicated structure out of these things over here. Perhaps we can actually reconstitute a real axoneme and actually make and understand better how axonemes are actually moving, but more work needs to be done to explain this. So let me go to the second part of the talk, which is about fuel for synthonemes, which is also related to the second talk today that we will hear from Magdeburg. We work together with the Magdeburg group. And so what Asam does is she took axonemes. So she doesn't take synthonemes like the ones we reconstituted. Here we take actually an axoneme, which is a demembraned, broken off flagellum of Chlamydimona. And when you activate it with ATP, then they're repeating, as you can see here. And so when you look at that, so what you do is you take this green algae, you cut off one of these little arms, you demembrane them, you put in ATP, you put a reconstitution system for ATP, and then they just keep beating. And you can see the imaging rate here is 1,000 frames per second. And over here, you see the time, they're actually beating much, much faster than the reconstituted ones that we were building before the synthonemes. And so if you look at the time evolution of one of these things beating along, what you can see here we have, it's very beautiful. I mean, I like this a lot, fits a lot to Santa Fe, by the way, in terms of the colors, I think. And so what you see here is you see these, the colors basically give the position of this axoneme in space as a function of time. So red is late times and blue is early times. And what you can see here is that the beating frequency depends strongly on the ATP concentration. So if the ATP concentration falls below a certain limit, you get no beating. And then if you increase the ATP, then you can see that the beat frequency goes up and they're beating faster and faster and faster. So all the way up to about 60 hertz, which is very, so one beat, 60 beats per second. So this is quite large. And so if you want to now do this, it's really tricky, because if I want to have, for example, my syntonemes or these exonemes to go forever, I need energy. So I need an energy module. I need to get some energy. And the one way we did this together with a group from Magdeburg, with Tanja Wiederowicz-Koch and Kai Sunmachte and Christine Kleinberg, what we used is we used biorobosyn, where we basically use light and have this as a proton pump, which then makes out of ATP. So we have this bacteriorhodopsin, and we have this synthase, and then they put this into a vesicle, and then they also studied how it depends, this energy production depends on the vesicle that they're being using, and so on and so on. But what you can see already is that the ATP concentration, if you have these vesicles, these lipid vesicles inside of a solution, you can see that the ATP concentration just increases. So just to show you that this works, you can turn on the light on and you can turn the light off, and what you see is that the ATP goes up, and then the ATP gets basically it decays because it's being removed, and then it goes in again, and so on. And so what you can do is you can say, well, what do we do? What happens if I put a lot of these vesicles in a solution, in a buffer solution where I have exonemes? Is it possible to actually drive these exonemes forever just by putting light into the system? The answer is yes. You can pretty much run these exonemes as long as you like, as long as you light the system and put light on it. So the idea is, again, that the ATP goes to your dyneins, the dyneins use this for their motion, and then the whole thing beats. And so here's a movie with this reconstituted system running. Azam and co-workers have investigated this very carefully, have looked at the time evolution, have looked at how long could they run this, and so on. And there's no apparent slowdown to be found. They also investigated different lipid vesicles and also the different structures, and have analyzed very carefully what the efficiency is and how it can be optimized. So then one question is, can we make something simpler? And so here Isabella and Azam come into play. And so the idea was, can we use this very simple system of two microtubules that we over here with the kinesin motors, put this whole thing into a small vesicle, which is made in basically a microtublet, which is in the oil phase. Then can we introduce this energy module over here, and can we then observe what should happen? So what we know is, if we do this in the bulk, we know that as soon as you give ATP to these motors actually will drive the microtubules in such a way that align themselves in a ordered structure into filaments. These filaments then very often make some kind of liquid crystalline-like structure, and the whole thing collapses like a gel. It's an active gel that is collapsing. Isabella Guido has a whole paper on this also. But let's see what happens in these little water and oil droplets. And so what you see is, you can see that that this network that is initially there on the illumination is contracting. So now we have to illuminate. We don't give actually external ATP, and then what it sees, you get also inside a flow, and you can see the structure over here. So this is after 0 seconds. This is after about 300 seconds. And what you see, the surface area has gotten much smaller. And what you see, there's this strong contraction depending on the size of the water and oil droplets that they have been using. And so here you see a video of the, well, does it? Yes, it's running, of the light-powered contraction of the microtubule network over here. So this is really light-powered. If you turn the light off, this whole thing just stops. And all this has been published, or is just appeared. It's not really appeared, but it has appeared in this ACS Synthetic Biology article. As you can see, there's the group of Kai Sunmachte, Tanja Wiederowicz-Koch, and many others over here which work together. Wahid is working here in our work with us. Albert is also Max Planck. Rahil is the graduate student. All this was funded from a EU grant, and they also got the front page of ACS Synthetic Biology. I think it hasn't appeared quite yet. You can see here it says XXX, but the paper is already online and you can read it up at this point. So I'm a few minutes early, but let me close with this. So here you see a picture of my group. So here's me over there. And so here are the most VIPs, the very important persons. So this is Azam and Isabella, Guido, who were running this investigation. And you can see I do many other things other than just synthetic biology. I'll be working on COVID-19. We're working on turbulence and transport of turbulent flows. We work on cloud microphysics, trying to understand why it's raining and why it's sometimes raining much earlier than anybody thinks it should. And so we work also on the Earth's climate, and we also work on cardiac dynamics and try to understand fibrillation in the heart. But this was not the topic of my talk here. This was all about synthetic biology. And with this, I would like to close. Thank you very much.
Thank you, Eberhard. That's a fantastic talk and some beautiful results. I want to again encourage the audience to submit questions through the Q&A. We have plenty of time for questions. The first question we have is coming from Jacqueline Delora, and she is asking, how is the beating of the synthonemes or isolated flagella dependent on the fluid dynamics of the submersion media? Is it important to reconstitute a physiological system, or in the implementation of a synthetic system, is it possible to diverge from what nature has already evolved?
Okay, so to be honest, what we do is all reconstituted, but I assume it's very close to biology, what's naturally there. Viscosity, probably you can tune as long as the osmotic pressures don't get totally berserk. But you should really ask Isabella and Azam that question. So I'm sure they're very happy to answer your questions. Just send them an email and they will come back to you.
Okay, thank you. Another question she had is, in the light-powered experiments, why are the droplets deforming?
Okay, so let's see. Do we know why? Let me see. So, well, hard to say, right? So what you see there is what you think what might be deforming. Let me go back one slide. So what you might think was deforming is you mean over here, this looks like, yes, it seems to be deforming. Well, very good question. Why would it be deforming? Because we have surface tension. I mean, this would mean this thing has to be anchored somehow to the interface, right? Otherwise, it cannot be deforming. Otherwise, we would assume that this thing is in the middle and would be surface. But they were deforming, so very good observation. I hadn't realized that here they are very round initially, as you can see, and this one is still round. But this guy here definitely looks like it's an egg. Fits very well to Easter, but it seems to be an egg shape. And so, very interesting question. But the other ones you see, so this is a very dense network. They're really right in the middle over here on the bottom. This one is deformed. But one would have to do experiments where these things are really not in contact with each other, with the other oil droplets, I would say. Yes, next question, please.
Next question comes from Susie Atlas, who asks, in the syntheneme work, are you planning to try to push further into a more fully biomimetic / artificial regime? And if so, which parameters do you imagine tuning or changing first?
Okay, so clearly I want to do that. So I got motivated by this idea of making a surface that is transporting fluids in a directional manner, even in some kind of lab on a chip kind of environment. So you could, at least the cilia in the brain, the ciliated ependymal cells that we have in the brain, they're very directional. They are evolving over time. There's directed transport. And this, of course, would be great, right? I mean, for example, have a spray would be fantastic. Somebody has a cystic fibrosis and you just have a little spray. A doctor came to me and said, don't you have a spray that I can spray to a patient and then he's healed? Well, this is, of course, would be a dream come true, right? You have a little spray and you spray in your artificial cilia, and suddenly the person has his own cilia and it's pumping again out the mucus and the person's safe. I think we're infinitely far away from that. But what is the next thing we would like? The next thing I would like to do is I would like to anchor these synonyms. And first of all, get a synonym that beats perhaps a little better than the one we have currently. But we already have axonyms, so you could also say but they're hard to handle. What would be great is to make an array of these and to anchor them and to make actually basal foot. I think the next thing that's needed is to anchor these to a surface and to make sure that they're beating in a certain direction. Because otherwise, how can you get so now we have to work on how to make a basal foot. This is my personal opinion is it might be able to do this with chlamydomonas or something and basically catch the whole animal and use them, but that wouldn't be a bottom-up approach. So I'm happy to hear any suggestions that you might have, but we're trying to capture these with antibodies or by mechanical means. It's not easy, and we haven't really made much success in that direction, but it would be great to make a ciliated carpet. So this is what I personally would like to do. I don't know what Isabella and Azam are after, but this is what I like a lot, is this ciliated carpets. Another one would be to make a vesicle and to attach some of these as a motor, so like a sperm, so have something that actually swimming along. And then to add to it some kind of chemotactic module that actually tells this object to beat in a certain way so that it's moving from A to B in a directional manner. That would be another thing that would be a high hanging fruit, in my opinion.
Thank you. We have a question from Felipe Quiroz, who's asking, what is known about the length of the flagella and its distribution in biological systems? Is the length a major and reasonable parameter to control in the context of artificial cells?
Well, I mean, the length that I'm aware of, they're typically, most of them are of the order of 10 microns. So the ones in my picture that I had, so the one that come from ependymal cells, flagella in sperm is longer, much longer. Then you get also this kind of wave-like motion. You not just have one, like in the chlamydomonas, you just have this one little beat. You never really have a full, you never have a long node, and then the whole thing is like a snake. What controls well, I don't know. What was the question again?
Is length a major and reasonable parameter to control in the context of artificial cells?
Well, I mean, length, I mean, it seems like 10 microns is something that comes about by itself. So you don't have to do too much to get something between 5 and 10 microns. It seems to be sufficient to get some beating. You could possibly make them longer. Is it a big factor? Well, it depends when you do the resistive force theory for fluid dynamics. So when you do the interaction of this object with the fluid, then length surely is a parameter that you have to consider. It will also determine the frequency and so on and so on. So the advantage of these synthetic systems is that you can really, at least we hope that we can basically take the system and make it as artificial as possible, so that we still have the major features, and then try to understand that system, and then go back up and say, what could the final system actually be? So the idea is a little bit what physicists love to do. They say, what is an elephant? What is a cow, right? And at the end is a sphere. So you want to go to the sphere and then work your way up to the more complex animal. But I think with the synthetic biology approach, we could do this. I should also mention, of course, that this whole field here of microtubules and motors goes into the field of active matter. So it very strongly also couples into the field of active matter. And so you get, active pneumatics, you get all kinds of different elastic properties. These are very different materials because, from the perspective of material science, what I show in the figure here, this gel that you see there, which is sitting in here, that's an active gel. This is not a passive gel, this is an active gel. And so these materials will be very, very different from anything we have been looking at in the past. Getting closer to biology, but perhaps also closer to really synthetic systems that have overlap with biology, but are not really biologically relevant, but might have other technical use.
Okay, thank you. We have another question from Lynn Rothschild, who writes, have you drawn any inspiration from natural biosynthetic pathways for cilia? For example, in the alga Chlamydomonas. Thanks, fascinating work, she says.
Chlamydomonas reinhardti. Yeah, so what we do is we harvest pretty much everything from chlamyd. So you know, we are growing chlamyd down in the lab. We have a little aquarium, and then basically things get harvested that way. Because you need you need the motors, you need the microtubules. We grow differently, but you might need the seed crystal, right? This is what I was showing here, where Isabella is growing the things out of basically a seed crystal, if you want. So when you go here, back here, right? So this artificial basal body, if you want, where you grow the microtubules out of this. So this is also from chlamyd. So this is stolen from chlamyd. So we are still using biology quite a bit. So this is not really fully bottom up. There is a little bit of use of biology, not just chemistry and so on. But in this case, this was the best. It's amazing that Isabella could do it that way. I'm still amazed that this works.
A related question is from an anonymous attendee who says, who asks, is the faster beating of the flagella from this Chlamydomonas compared to the synthenemes a function of the number of molecular motors and number of microtubules involved?
I would say this is a very interesting question. Very interesting question. Well, clearly, I mean, I would suspect yes, but this is now a pure guess. It's much faster, right? It's much faster. I mean, 60 hertz is quite a speed, to be honest. And we have, of course, multiple of these structures that could give you an increase in frequency, right? If you interleave these in such a way that some basically are recovering while the other one is contracting, they could give you an increase in frequency. I don't know. Something I have to think about, but a very interesting question for sure. Thank you.
Sure. Well, thanks to the anonymous attendee. Okay, we have one more here. And I think maybe you've addressed this, but I'll go ahead and ask it. It's from Ana Relimpio. She asks, did you try reconstructing the microtubules in the axonemes of the algae, only cutting its end?
Oh, she wants to, I see, she would like to grow out at the end. Talk to Azam and Isabella.
Okay.
I think the control is not quite that good, but I might be wrong. So please talk to Azam and Isabella.
Okay. Well, I would encourage anyone in the audience who has further questions to definitely email the people that Dr. Bodenschatz has mentioned, and also Dr. Bodenschatz, if you have any questions.
Just send me a copy so I see what you asked.
Okay, Eberhard, well, thank you very much for the very interesting talk. I will introduce the next speaker in just a few moments here.